Unveiling a revolutionary discovery, a drug to overcome cancer's resistance to chemotherapy. In the intricate battle against cancer, our cells are not alone in their fight. Cancer cells, with their cunning disguise and manipulative tactics, thrive by deceiving other cells, even manipulating immune cells called macrophages into building protective barriers around tumors, rendering treatments ineffective. However, treatments can unite to overcome this challenge. Researchers at King's College London, KCL, have developed a groundbreaking companion drug in the form of a daily pill, designed to dismantle these immune barriers, enhancing the effectiveness of chemotherapy and offering hope to more patients. The initial laboratory study of this drug, KCL-HO1I, has demonstrated its potential. It has shown remarkable results in mouse models, successfully making chemotherapy-resistant tumors vulnerable once again. We uncovered that macrophages act as guardians of the tumor, blocking the effects of chemotherapy, explains Professor James Arnold, head of the Tumor Immunology Group at KCL. They act as gatekeepers, preventing cancer-fighting immune cells from supporting the treatment. But by targeting the right pathway, we can unlock the door. Arnold and his colleagues, supported by the Medical Research Council, have established Ethox Therapeutics to bring this drug to clinical trials. Breaking down the tumor microenvironment. The development of KCL-HO1I has been a long journey, spanning almost 13 years. Professor Arnold's lab focused on studying the role of macrophages within and around tumors, known as the tumor microenvironment, in the early 2010s. Early observations revealed that these tumor-associated macrophages produce an unusually high amount of an enzyme called heme oxygenase 1, HO1, leading the team to delve deeper into its function. It's an enzyme that has become almost like a family member, Arnold shares. I've spent countless sleepless nights trying to unravel its mysteries. Their efforts paid off. The research team discovered that HO1 acts as a shield, protecting tumors from the body's natural defenses. What we found was that these macrophages and heme oxygenase 1 play crucial roles in regulating the immune response against cancer, which significantly impacts the efficacy of chemotherapy, Arnold explains. This revelation came as a surprise, as chemotherapy, which targets fast-dividing cells, is often considered separate from the immune response, which identifies and targets abnormal cells. Immunotherapy, a specific treatment designed to enhance the immune system's ability to recognize and destroy cancer cells, further complicates this picture. However, Arnold's team found evidence that the most successful chemotherapies also seem to harness the immune response against cancer, primarily by activating potent cancer-killing T cells. While the exact mechanism remains to be fully understood, we now know that when chemotherapies initiate the killing of tumor cells, they can activate T cells to join the attack. It's a powerful 1-2 punch, but for this strategy to work, T cells must gain access to the tumor. This is where macrophages, responding to different signals, pose a challenge. The macrophages believe their role is to protect the cancer, so they produce heme oxygenase 1 to keep T cells at bay, Arnold explains. When we targeted heme oxygenase 1 using KCL-HO1I, we observed a significant increase in chemotherapy's effectiveness, primarily due to a stronger immune response against the cancer. The immune system, evolution, and cancer. Chemotherapy, one of the most established cancer treatments, only gained prominence in the 1960s. The problem addressed by the KCL team has deeper roots, stemming from millions of years of evolution. Throughout human history, most individuals did not live long enough to face age-related diseases. As a result, the immune system has evolved to protect us from problems common in younger ages, either by ramping up to fight infections or calming down to support wound healing. In today's world, where people are living longer, we encounter situations where neither of these responses is entirely appropriate. The immune system simply lacks the tools to effectively combat a disease like cancer, Arnold says. Unlike the pathogens that cause infections, cancer cells originate from our own bodies, growing and dividing uncontrollably. With each division, they have the potential to evolve new disguises. To macrophages, a tumor producing new cells may resemble a healing wound rather than a threat. Macrophages support wound healing by blocking access to certain parts of the body from T cells, which can disrupt the healing process. They produce heme oxygenase, one to provide space and time for wounded tissue to recover, but this can inadvertently aid cancer growth and spread. The immune system is essentially misinterpreting the situation, leading to catastrophic consequences, Arnold explains. Our therapeutic approach essentially reconfigures the microenvironment, allowing T cells to enter. It's removing the tumor's defenses. From discovery to drug development. While Arnold's lab identified a long-standing problem, transforming a discovery into a new cancer drug presents a unique set of challenges. Fortunately, they didn't have to start from scratch. Other researchers were already testing a heme oxygenase 1 blocker as a treatment for jaundice in newborns. This provided the team with the knowledge that drugs could safely and effectively target this cancer defense system. However, the jaundice treatment is designed as a one-time hospital infusion, whereas a chemotherapy companion drug would need to be modified for repeated use. This is where Arnold's KCL colleagues, James Spicer, Professor of Experimental Cancer Medicine, and Miraz Rahman, Professor of Medicinal Chemistry, stepped in. By combining Rahman's chemical expertise and Spicer's clinical knowledge, the team developed KCL-HO1I, a new drug that can be administered as a daily pill. This means that individuals receiving chemotherapy could take it at home between treatment sessions, potentially enhancing the effectiveness of hospital treatments without requiring additional hospital stays. A collaborative effort to improve chemotherapy. This achievement is a testament to the power of teamwork across different disciplines, Arnold says. We recognized the potential for a new drug opportunity for cancer patients, which motivated talented individuals like Dr. Mariam Bari, the lead author of our paper, to contribute to this journey. The paper truly encapsulates all our efforts, and we are thrilled to be in this position. The next step, which Arnold believes is achievable within 2 years with the right funding, is to begin testing the new drug in clinical settings alongside established chemotherapies. There is a wide array of approved chemotherapeutic drugs that are widely used, he explains. We call this a companion drug because, in theory, it could benefit any patient receiving chemotherapy, allowing for a better treatment response. Initially, they plan to work with standard doses of the chemotherapy drug gemcitabine, but if they can boost responses, it could mean that doctors could opt for milder chemotherapy treatment plans. It's an application of evolutionary principles to some of our most common cancer treatments. We have much to explore, Arnold concludes, but in the future, we could see a range of combination cancer treatments that include KCL-HO1I. This groundbreaking research offers a glimmer of hope in the ongoing battle against cancer, showcasing the power of scientific collaboration and innovation.